And without further ado, Patient-Centered Care, May 8th, Dr. Lori Ramkissoon, From Bench to Bedside, Molecular Oncology's Role in Personalized Cancer Diagnosis and Treatment. All right, I'll put up the more extensive bio in case anyone would like to read that. Dr. Ramkissoon, thank you so much for being here today. Welcome.
Thank you for inviting me. I appreciate the opportunity.
Absolutely. So we've got a little bit of information about you here. Director of the Cytogenetics Laboratory at University of North Carolina Medical Center, and earned her PhD from, help me pronounce that, is that Weill or Whale?
Weill Cornell.
Weill Cornell Graduate School of Medical Sciences, and did a postdoctoral fellowship at Dana-Farber Cancer Institute. And prior to graduate school, a predoctoral fellow in the laboratory of Dr. Neil Young at the National Heart, Lung, and Blood Institute in Bethesda, Maryland, and worked for a year as a staff assistant in the United States Senate. All right, that's a great background.
Which encouraged me to go to the National Heart, Lung, and Blood Institute and be around people who I could follow their thought processes a little more clearly. So.
Gotcha. I love hearing about people's career trajectories, so thank you. What's something else we should know about you outside of your professional bio there?
Well, at the end of June, beginning of July, my family and I are looking forward to our first trip to Seoul, Korea.
Wow.
So I'm very excited about that vacation.
Oh my gosh. Well, we hope you have a great time. And that's all fun?
All fun.
Good for you.
Yes.
For you. All right. Well, I mentioned Poll Everywhere. That question should be popping up for everyone who already went to Poll Everywhere. And the question is, cancer is fundamentally a disease caused by changes to the normal sequence of a patient's genome. And the goal of molecular oncology is to define and understand these changes to benefit the diagnosis and treatment of cancer. That'll be a true-false question. I'll put up our ACCME disclosure and our ANCC disclosure. And I see that we already have people answering that question. We'll give you about 10 more seconds. Take your best shot. I think I forgot to mention that everything with Poll Everywhere is completely anonymous. So we're not going to come back and talk to you if you got the answer wrong. This is all anonymous, but we do appreciate your participation. And I always suggest folks jot down questions as we go along, and then you can go ahead. We'll let you know when you can start typing those into Poll Everywhere later on in the presentation. Oh, so how did they do?
We have a very savvy audience already.
Great.
So
great.
Already know that this is going to be great.
All right. Well, from bench to bedside, molecular oncology's role in personalized cancer diagnosis and treatment. Let me turn the controls over to you.
Okay.
It's the keyboard, and there's the mouse, and we do have the cursor working on the mouse too, if you want to use that.
Okay. Well, since everybody did so well on that Poll Everywhere question, how about we try another one? Another true-false. Molecular biomarkers are only used in the research setting and are not part of current standard of care clinical paradigms. Again, I think we're working with a very, very savvy audience today, so that's great. It's false. Molecular biomarkers are actually an integral part of clinical care. And like we mentioned in the test Poll Everywhere questions, most cancer mutations are acquired during a patient's lifetime rather than inherited from our parents. And there can be many reasons that a person over their lifetime acquires mutations. They can be caused by environmental factors like tobacco smoke, UV radiation, or just random errors that occur during DNA replication as cells divide. So the goal of molecular oncology is to leverage advanced technologies like genomics, transcriptomics, and other biomolecular techniques to identify and understand how changes to the cancer genome drive the oncogenic process. And by understanding and applying the molecular understanding of the tumor, we can tailor personalized target strategies for patients to improve their overall survival, to monitor their response to therapies, and really try to improve overall patient outcomes. So over the next hour, my goal is to review advancements in laboratory methods used to detect molecular biomarkers, and then use a couple disease examples to illustrate how molecular biomarkers have been integrated into diagnostic algorithms. And then finally discuss how we use these molecular biomarkers in different treatment strategies for cancer. So before the advent of molecular targeted therapies, we had three basic cancer treatment primarily relied on surgery, chemotherapy, and radiation. And these tools are still an important agents in cancer treatments today. As the first line of treatment, often, especially for solid tumors, the goal of surgery is to remove cancerous tissue and any affected areas around it with the goal to eradicate or prevent the spread of disease. Surgical resections or biopsies, fine needle aspirations, any procedure to acquire tumor tissue is also important in molecular oncology, as this gives us the samples that we can use to test to figure out what the changes are of that cancer genome. Chemotherapy, on the other hand, uses cytotoxic drugs to kill cancer cells or to stop them, to prevent them from rapidly dividing. However, these are often non-specific, and since their goal is to attack dividing cells, they really don't discriminate between malignant cells and healthy cells. So this can lead to a variety of different side effects, and may kill the tumor, but also may do harm to our normal healthy cells. Some chemotherapy can be systemic and affect the whole body, or it can be targeted now. It can also be used before surgery or after surgery or in combination with radiation. And radiation is an important method that uses high energy rays or particles to destroy cancer cells. It can be delivered externally or internally. And again, it might be used before surgery to shrink a tumor. It might be used after surgery to eradicate those tumor cells that weren't able to be eliminated with surgery. Or it can be used as a standalone treatment for those cancers we know can't be extracted via surgery. The goal of precision medicine ushered in a new form of therapies that now make things more specific to tumor cells. And it gave us a lot of different treatment options that can be used in combination with the conventional treatments that I discussed. These new therapies include targeted small molecules, monoclonal antibodies, and immunotherapies, some of which will go into more detail on how they apply to specific cancers. But the combination of these precision medicine agents have really advanced how we treat advanced cancers. So when we think about molecular biomarkers and how they contribute to cancer, it's really a spectrum throughout the entire process about how molecular biomarkers can inform a patient's journey. Before cancer, some families have a long history, so there are certain markers that we can test for to determine if they're at an increased risk for cancer. Additionally, if they have a diagnosis, more and more molecular biomarkers are used to further define or classify that tumor. After a patient has received a cancer diagnosis, molecular biomarkers have a variety of different roles. We know from retrospective studies that if a certain genomic profile of a tumor maybe has a different prognostic trajectory than other genomic profiles. We also know that there's certain genomic changes or molecular biomarkers that we can predict response to a treatment. We also know that a person's genotype can also affect what kind of dosing they should get for certain types of drugs. The study of pharmacokinetics, which we won't go into much today, but is another example of a molecular biomarker. And finally, advancements in our technologies to detect genomic changes. We can then use this to monitor the treatment course, how patients are responding, and whether the tumor is changing and becoming resistant to certain treatments. There's also fields of study that look to see when or if a tumor will recur. So really, biomarkers are an important part across the entire spectrum of the cancer journey. So let's talk a little more specifically about molecular biomarkers in cancer. When we talk about biomarkers, what how are we defining that? Generally, a biomarker is considered a cellular, biochemical, or molecular alteration that can be measurable in human tissues, cells, or fluids. So we can think of blood pressure as a physiological biomarker, or we have things like C-reactive protein that is an inflammatory biomarker. Essentially, biomarkers can be used to screen diseases, distinguish between malignant and benign processes, again, predict response to therapy, determine which therapy a patient should get, monitor a patient's status, and really also address whether they're at risk of other diseases or cancers. But today I'm going to focus more on the molecular or somatic mutational biomarkers as they relate to cancer. So how are molecular biomarkers used in oncology? There's really three overarching concepts where molecular biomarkers play an important role. The first is diagnostic. Again, as we learn more about cancer genomes across a variety of different tumor types, we're able to use genomic profiles to be a little more specific about the type of cancer a patient has. For example, some molecular changes are diagnostic. For instance, the BCR-ABL gene fusion is specific for chronic myeloid leukemia. They can also offer prognostic significance, essentially determining if a patient's disease will behave more aggressive or less aggressive compared to other patients with similar cancer types. This can be seen in p53 mutations in chronic lymphocytic leukemia. We know that those, if present in a patient's disease at diagnosis, are an adverse prognostic factor. And finally, they are becoming increasingly important as determinants of which therapies might be best for the patient's tumor. They can assist not only in choosing which treatments patients will respond to, but can also be indications as to which treatments patients should not receive because they have a resistance mutation. And more molecular biomarkers are used to establish clinical trial eligibility, which, as we know, is an important factor in advanced cancer patients. So when we look at the integration of biomarkers in cancer, when we look at the NCCN guidelines, there's over 800 biomarker recommendations to date, and these are rapidly expanding. Each update, there's different biomarkers, but being added to the NCCN guidelines. But again, these can be things like screening for BRCA1 and BRCA2 mutations to determine the risk of patients' risk for disease. They can be diagnostic, prognostic, they can be predictive. Again, certain pharmacogenetics biomarkers can be used to determine the risk of toxicity. It's really an expanding and continuously evolving field, but it has been integrated into our guidelines for how we treat patients. When we look specifically at one tumor type, non-small cell lung cancer has really seen significant advancements in how molecular biomarkers have transformed the treatment of this tumor type. In 2004, the first oncogenic EGFR mutations were detected in non-small cell lung cancer. Now, decades later, the spectrum of molecular biomarkers that offer targeted therapies or resistance or some sort of prognostic information has really expanded significantly. And these biomarkers have revolutionized in non-small cell cancer lung treatment. And now the guidelines, NCCN guidelines and other professional guidelines really recommend that all patients with advanced stage lung cancer be screened because of the significance of these biomarkers in treatment selection. Another tumor type that has where biomarkers play an important role is colorectal cancer. It's the fourth most frequently diagnosed cancer and the second leading cause of death. And molecular biomarkers play an important role, for example, in patients screening for loss of mismatch repair proteins, which can be an early indication that they need to be screened for Lynch syndrome. Lynch syndrome is a hereditary cancer predisposition syndrome where a lot of the patients get colon cancer, but it's important to identify these patients as they're also at risk for other forms of cancer. So it's really important to identify these patients not only for their risk of future cancers, but they may be eligible for a specific type of immunotherapy. Additionally, the NCCN strongly recommends for all advanced colorectal patients to screen for KRAS or the RAS family mutations. As these patients should not be treated with an EGFR inhibitor, since we know that they do not respond to these types of inhibitors. And increasingly, other biomarkers like HER2 amplification and BRAF V600E status is becoming important to know in advanced stage colorectal cancer as they offer specific targeted treatment options. So colon cancer and non-small cell lung cancer were just a few examples, but as you can see from this schematic, really almost all types of cancer have molecular biomarkers that are associated with some form of critical information that provides patients, whether it be for their treatment options, for the prognosis and how aggressive maybe they should be with treatment. And you see some overlapping themes of genes that are involved in different tumor types. And this should come as no surprise, as we know in cancer, the goal is for them to rapidly divide, turn off tumor suppressive mechanisms. So a lot of these genes play significant roles in signaling pathways that drive this proliferation. But we do know that each cancer, even if it has let's say a BRAF V600E, behaves a little differently and may not always respond the same as another tumor type that has these mutations. And what's exciting in the molecular oncology field is now there are tumor agnostic biomarkers in solid tumors. So it doesn't have to be a certain tumor type, but if you find an NTRK fusion, we know that now that biomarker alone can get you a targeted therapy. Other things like tumor mutation burden and microsatellite instability are now biomarkers that, regardless of your tumor type, have associations with treatment. Because molecular biomarkers play such a key role, the American Society for Clinical Oncology, or ASCO, has established guidelines that recommend that patients with metastatic or advanced solid tumors, if there's a genomically approved biomarker linked to a therapy, that these patients should be receiving molecular testing to identify biomarkers. And again, now that we have tumor agnostic therapies available, this really applies to all advanced solid tumor patients. Again, patients with metastatic or advanced solid tumors, if there's clearly defined resistance markers, these should be identified before treatment is being considered. So really, molecular biomarker testing should be standard of care for advanced solid tumor patients. So in talking about detecting molecular biomarkers, that's great, but exactly how is this done and what are we looking for when we talk about genomic changes? So here I just wanted to talk a little bit about what we mean by genomic alterations and what they mean in the oncology context. So there's really different types of changes that can occur to our genome. There's things like base pair substitutions. So this is limited to a single nucleotide in our DNA sequence. And it can be anything from a missense change, where it changes one amino acid to another. It can be a change to the nucleotide that now all of a sudden creates a stop codon where there shouldn't be. We also see single nucleotide changes can result in a reading frame shift so that it no longer encodes for the full protein and essentially creates a truncated mRNA that doesn't get translated into a protein. So a single change to our DNA can have significant disruptions. We can also have copy number alterations to our genome. This can occur by gaining more and more copies of a genomic locus or a specific gene to essentially have more of that protein that the cells use to drive proliferation. Examples of this are HER2 amplification or PDGFRA amplification. We can also see what we think about a little more complex changes to the genome by insertions and deletions. In this case, instead of a single nucleotide being affected, maybe we're inserting more DNA bases into the sequence to, again, which will affect it by throwing off the reading frame or introducing in another string of amino acids that changes the protein conformation. We can also see small deletions within a gene that can disrupt the normal function. And examples of these can be EGFR exon 19 deletions or MET deletions. And finally, there are also genomic rearrangements. So we've known that even though our genes are segregated to different chromosomes, we know that there can be changes from location to location. Certain genes can come together to create novel oncoproteins that drive proliferation. These can either be detected by RNA or DNA testing. And examples of this include ALK fusions and INTRAC fusions. And it really depends on what the genomic change does in the cancer cell that becomes important. And when we think about genomic changes, we think about driver versus passenger alterations. So when we think about driver alterations, those are changes to the cancer genome that give the cell a competitive advantage compared to the normal cells within the tissue. These genomic changes tend to increase the rate of proliferation. They tend to oppose apoptotic mechanisms that our cells have to take care of changes that could be detrimental, or they could provide resistance to therapy options that patients have. Essentially, driver mutations we think of as they contribute to oncogenesis. There can also be passenger mutations. So they are just kind of along for the ride and don't necessarily give a proliferative or competitive advantage to the tumor. These are types of genomic changes or mutations that arise in cancer cells, but they don't hurt the cancer cell, but they don't also give it that competitive advantage. So it's important that we be able to distinguish what is driving the tumor and what is kind of along for the ride. And when we think about the purpose of genomic changes in cancer, we think about gain of function versus loss of function alterations. And depending on the function of the gene within the tumor and then what type of alteration is needed in that gene to give that cell a competitive advantage can drive kind of how we think about treating or targeting that alteration. So targetable alterations that are gain of function are usually mutations that occur in oncogenes that encode for signaling molecules. So signaling molecules that can drive pathways that are important for our cells to grow and divide and perform important functions for that tissue. These gain of function mutations are frequently heterozygous, meaning that only one copy of the gene needs to be altered in order to have this competitive advantage. And for gain of function variants, there's really just generally a limited number of mutations that are needed to activate a protein. Usually gain of functions tend to be recurrent among individuals. So, for example, in myeloproliferative neoplasms, a very specific single missense change at one location, as demonstrated by this image of a cancer database where we can look at the spectrum of mutations across, we see that the vast majority of mutations in the JAK2 gene occur at one codon. That's because the cancer cell can identify a key component of that protein mutated, and it has a competitive advantage. So gain of functions tend to be similar among different tumor types. Loss of function mutations, on the other hand, can typically occur in tumor suppressor genes. And for these type of mutations, the loss of function needs to wipe out both copies of the gene instead of just activating one. So we need to inactivate both copies to eliminate the normal protein function. And because we're looking to suppress or lose the function of that gene, there can be many different alterations across the sequence of the gene that can result in a loss of function. So as demonstrated with the RB1 gene, in order to inactivate that protein, we can see that there's a spectrum of mutations all across the coding sequence to inactivate that. So this kind of matters in depicting what type of assay we're going to need to use to identify important alterations in tumor. So part of understanding molecular biomarker testing is determining which assay is right for the question that you're trying to answer. So this kind of lays out some questions that as a laboratory professional we ask, and to try to answer the questions that are needed with the amount of tissue or material that we have to work with. So does the assay cover the required genes and portions of genes? For those oncogenic gain-of-function mutations, maybe we just need to target one codon of that gene, and that's all we need to know because that's only where the oncogenic mutations have been detected. If we're looking at tumor suppressor genes, we need to look at the entire gene to make sure that we don't miss any loss of function mutation that can occur throughout the gene. We need to ensure that the assay can detect the types of variants, whether it be those single nucleotide variants, those changes that affect only a single base pair, or if we're looking for larger genomic changes, larger gains of genomic material or loss of material, that may require a different assay. When we're talking about gene rearrangements, we need to understand if we should be looking at just RNA to look for the fusion transcript, or if a DNA-based testing method can detect recurring rearrangements. Also, what's becoming increasingly important is what are the sample requirements? For some testing, the fixation of tissue or how the tissue is processed matters. Things that have been decalcified, so like bone metastases, anything that requires a more significant process in order to look at the tissue can affect how the genomic technologies that we use to interrogate the DNA. We can also think about how much tumor cells need to be within that sample type. And additionally, what is the expected turnaround time and cost? Because certain molecular biomarkers provide critical information before treatment, maybe we need to find an assay that gives us a faster turnaround time than a more comprehensive test. So all of these factors need to be considered when we think about whether we're looking for a single variant, which may be one type of assay, or if we need a more comprehensive look at more genes, more types of variants, with the understanding that the more of the genome that we need to look at, the longer of a turnaround time and essentially, or increased costs this can be associated with. So what are some of the technologies that we use in the laboratory to detect molecular biomarkers in cancer? So we can start with the more broad, kind of higher level things like chromosome analysis. This is the methodology that we use in the cytogenetics lab. And this is a really valuable tool to detect changes in chromosome number or translocations where part of the genome moves to another part of the genome. We can also look at large deletions insertions. This is essentially we're doing a whole genome analysis in 20 cells, but it's really like high or low resolution. We're, an airplane view above the genome when we're using chromosome analysis. So we're really looking for large changes to the genome. If we know that we're targeting something specific, a specific genomic rearrangement, or a specific deletion, we kind of know what we're looking for. Fluorescence in situ hybridization, or FISH, is a great method to be able to detect some of these changes. These methodologies can have a rapid turnaround time. Again, if we know what we're looking for, they can, in some instances, we can have a result within a day. SNP microarrays are useful for genotyping and also copy number determination. This provides a little higher resolution for copy number changes, so gains or losses within the genome compared to chromosome analysis. If we're using a single nucleotide polymorphism microarray, we can also use this to look for loss of heterozygosity. So you may have two copies of a gene or a region of the genome, but we've lost the heterozygosity, or the contribution from our parents, and now it's essentially the same, only information from one copy of the gene, which can have prognostic information significance in some tumors. There's also gene expression arrays. So instead of the DNA sequence of the tumor, what kind of mRNAs or what kind of genes are being expressed that drive a certain phenotype? We know that certain gene expressions can subtype tumors and have prognostic significance based on those expression profiles. Additionally, if we want to look at a little higher resolution, now get down to the individual base pairs of a tumor genome, we can use single gene assays. So again, this is just looking at one gene that we know is important in a tumor type, and we can evaluate for those single nucleotide variants. We can look for small insertion deletions, 20 to 50 base pairs in size, and really can be very targeted and give limited information. Another important tool that now we're looking at protein expression is immunohistochemistry. This defines protein expression within a tissue sample. And this is important for, that's the functional readout of what our DNA encodes for. And we can determine in certain specific cell populations which cells are overexpressing or have that protein marker. So this is an important tool in our cancer diagnosis. We can also combine single gene assays and look at multiple genes and sometimes what we call hotspot panels. So now this is looking at multiple genes that are important in cancer, but only looking at specific regions of those genes to identify important biomarkers. This can be more efficient if we know what variants in specific genes that we're looking for. So it can provide the specific information that we need and not any other kind of changes that we may then have to decide, is that contributing to oncogenesis or just a passenger mutation? But more and more, we are using broad panel sequencing or comprehensive genomic profiling, which is a next generation sequencing test that looks at it can be a defined list of hundreds of genes, or it can be the entire genome, but we're looking at a lot more mutations, a lot more genes, and trying to parse out which variants are critical for oncogenesis and which are passenger mutations. The comprehensive genomic profiling can be performed on DNA or RNA testing. So how do we know which methodology to use and for which tumor type? And fortunately for some cancers, the NCCN helps us, as there are guidelines that define the type of assay that we should use and their significance. For instance, HER2 biomarker testing in invasive breast cancers. The guidelines recommend that we start with a HER2 antibody and define the level expression in those tumors. Depending on the IHC score, this may dictate whether the patient's tumor is HER2 negative or HER2 positive. As you can see from the IHC images there's a spectrum of protein expression from a 3 plus, which indicates the tumor is strongly expressing HER2, to a 1 plus, where we do not see much expression of HER2. Now, when we have that middle 2+ where it's not strong staining, but there is some, the guidelines suggest that we look at another method to determine if HER2 amplification is present. And that's where fluorescent in situ hybridization, or FISH, plays an important role. So for HER2 IHC 2+ tumors, the guidelines recommend that a reflex FISH test be performed to determine at the genomic level whether there's an increased number of HER2 copies. And we use this by using a probe specific to HER2 and then control for the copies of chromosome 17 using a centromere probe. And by using these two methodologies and based on the guidelines, we know that if the ratio of HER2 to the centromere is above a certain number of copies, that this can give the tumor a HER2 amplified status, which can then make them eligible for a HER2 targeted therapy. But for some tumors where we have a lot of different biomarkers, a lot of different variants, we're kind of looking maybe also for a profile of the tumor to help us determine the origin of the tumor. Really comprehensive genomic profiling can be the best option for detection of molecular biomarkers. So just to review the process, tumor DNA is extracted from the sample, and then it needs to be chopped up into smaller pieces or fragmented. And then it goes through a process called library preparation, which essentially just means that different sequences need to be added to those fragments such that we can then direct a polymerase to make multiple copies of that segment of the gene. We also put barcodes on it so that we can run multiple patients on one assay and then bioinformatically tease apart which patient had which kind of variant. So that's kind of all part of the library preparation. And then because for most comprehensive genomic profiling in somatic tumors, it's a panel of hundreds of genes. So we don't need some of the other DNA that does not have any significance to us. So we enrich for only those targets that are important. Then once we have our enriched library, the DNA is attached to a solid surface and the sequencing process can begin. Now, the process of sequencing happens, and now we have all this data that must go through different levels of processing so that we can take the sequences that were generated from multiple individuals' tumors, align those to a reference genome, and then compare the tumor genome to the reference genome. So there really requires a significant bioinformatics process. And once we have our list of genomic changes that were present in the patient's tumor, then we need to go through and determine, is this biologically significant? Does this give some indication that the patient is eligible for a specific therapy? Is this a resistance marker? So classification of the variants is also an important component of next generation sequencing. So the benefits are it's really high throughput. You can get a lot of patients' genome or important molecular biomarkers assayed with a single test. It can address all targetable, actionable regions of the genome, but it really requires a huge infrastructure cost. Not every hospital lab, institution, or even reference lab can support the infrastructure needs that next generation sequencing requires. And because of the comprehensive nature, there is a longer turnaround time associated with comprehensive genomic profiling. So when we think about the comparison of a single gene testing algorithm versus a comprehensive genomic profiling, there's really some things to think about that can maybe determine whether single gene testing is more effective than comprehensive genomic profiling. PCR and single gene testing, again, there's more options for how we look at the genome and can identify certain markers, but typically it's it's performed in a reflex kind of action. You start with one gene, and if a variant or the biomarker is not present, then you move on to the next gene. So this can require a lot more time and resources that are needed to evaluate all the biomarkers that are important. Also, for comprehensive genomic profiling, again, you can get a look at all the biomarkers by DNA, RNA sequencing, and now it can also provide information for those tumor agnostic indicators. One key thing that's important to also determine is what is the material that we have to work with. We kind of think about two options. We either have a tissue biopsy or a liquid biopsy. And tissue available for testing is still the gold standard for determining the genomic profile of solid tumors. And this can be done through tissue resection, biopsies, or fine needle aspirations. But this is, again, limited. Some patients are not fit for resection, or biopsy is not feasible. So while this is still the gold standard, sometimes it's just not feasible for all patients. But it is important to understand that tissue biopsy can also give us information about the tumor rather than just the genomic profile. So tissue-based testing is still really important in cancer diagnosis. But liquid biopsy is becoming an increasingly important source of tumor genomic profiling. And what we mean by liquid biopsy is taking the circulating tumor DNA, which is a component of the kind of stuff that is flowing through our blood in addition to our blood cells, and use this to evaluate the tumor profile. So we know that tumors shed their DNA into the bloodstream either through apoptosis or there's necrosis, a variety of different mechanisms that can release circulating tumor DNA into the bloodstream. And there's specialized tubes that this biopsy should be collected in because these fragments have a short half-life. So we need to try to stabilize them so that they can get to the facility or laboratory for testing. And studies have shown that the amount or prevalence of ctDNA can vary among tumor types. The study looked at a variety of different tumors, and you can see for like bladder cancers, colorectal cancers, for all the patients that they tested, they were able to identify circulating tumor DNA. But we know that there's some tumors like brain tumors or thyroid cancers where there's just not that much circulating tumor DNA that's shed into the bloodstream. We also know that the level of circulating tumor DNA can vary between cancer types. Colon cancers tend to have a higher percentage of circulating tumor DNA than other types of cancers like ovarian cancer. So while this liquid biopsy is a great alternative, we also need to think about the tumor type and that circulating tumor DNA is not equal between tumor types. When we look at the concordance between liquid biopsy and tumor tissues, studies have demonstrated that there is a high concordance for most tumor types between a liquid biopsy and testing tumor tissue. When we look at non-small cell lung cancer, we can see that there's approximately a 60% concordance rate between tumor tissue and liquid biopsy. And we can also see that there's unique variants found in circulating tumor, the liquid biopsy compared to the tissue biopsy and vice versa. And the study found that the more circulating tumor DNA that you have, the more unique variants can be identified. And this can be important because we know that the circulating tumor DNA is more reflective of the heterogeneity of the tumor than a tissue biopsy can be. So there's advantages and disadvantages to tumor and liquid biopsy, and we must weigh the advantages and disadvantages. But it's also becoming important to know that a false negative on liquid biopsy should prompt a tissue resection or a tissue biopsy, as we know that different stages of tumors or different responses can affect the level of circulating tumor DNA. But for liquid biopsies, there are also disadvantages where we identify variants that may not be associated with the cancer, but can be indicative of other processes like CHIP or clonal hematopoiesis of indeterminate potential. So there are guidelines recommending liquid biopsy if no tissue is available or if the tumor is aggressive. So it's really becoming an integral part of how we think about molecular biomarker testing in cancer. So gone a little long. Let's go to our second question. Which testing methodology will most efficiently detect biomarkers in lung cancer? And here's the recommended testing from the NCCN guidelines on the right.
We'll move to if you can move to the next slide, I think that one, there we go.
Okay.
That's got the poll everywhere. We've got A, individual FISH assays to integrate each biomarker, B, hotspot DNA gene sequencing panel, or C, DNA and RNA sequencing assay. Well, this one's challenging.
This is challenging. And, you know, I think when we think about the recommended testing, from my perspective, I see that there's all different types of genomic alterations that need to be evaluated. So really a DNA and RNA sequencing based assay is the best method for testing in lung cancer, as it'll identify rearrangements, mutations, and different alterations. Given the number of genes that have associated therapies associated with it, one study the guidelines actually recommend that molecular testing that includes all these genes be conducted before first-line therapy. And these guidelines do offer some guidance that, again, which testing should be performed, and then how we can use that information to guide treatment strategies. And this study actually showed in 300 lung patients that it's really critical to get the comprehensive genomic profiling in non-small cell lung cancer, as the survival is actually affected by whether patients get all biomarker testing upfront or do not get any testing or even incomplete testing. So while FISH can identify some of those, the comprehensive genomic profiling has been shown to be more sensitive and accurate in detecting biomarkers in lung cancer. So now molecular biomarkers are also refining how we classify tumors. And this is illustrated, I think, best in hematologic malignancies. Back in the early 2000s, we used this French-American-British classification system that really relied on morphology, flow cytometry, and immunohistochemistry. But as we got to learn more about the genomic rearrangements that are present in different tumor types, we started to see specific classifications of acute myeloid leukemia with specific genomic rearrangements. And the classification of tumors has evolved to include these specific type of genomic rearrangements. And now you can see the number of genomic rearrangements has expanded in the current version. And what's important is that by identifying some of these rearrangements, you don't have to have the morphologic features or a certain blast percentage in order to get a diagnosis of AML. And this matters because we know that certain genomic rearrangements are associated with a worse overall survival. So again, they can be diagnostic without having the morphologic or certain blast percentage, and can provide an opportunity for the appropriate therapy. And in AML, it's important to understand which risk category a patient fits in as to determine whether they should receive intensive care chemotherapy or will be a better candidate for a stem cell transplant and or targeted therapy. So really genomic rearrangements and now mutation profiles are becoming diagnostic in certain subtypes of acute myeloid leukemias. We've also seen molecular biomarkers increasingly incorporated into neuro-oncology. Again, it used to be based on just morphology, presence of certain morphologic characteristics. But now we see an evolution from the 2016 WHO to 2021, where again, certain genomic rearrangements are diagnostic of subtypes of gliomas. And you can follow a flow chart where, again, if you have certain molecular biomarkers without morphologic features, it can give you a high-grade diagnosis, which can, again, affect how patients are then treated. So genomic biomarkers are becoming increasingly important in the diagnosis for many different cancers. And I'd just like to spend the last few minutes on how biomarkers can guide treatment decisions. Again, an example of lung cancer, how over the past 20 years, we've seen a significant increase in the number of targeted therapies available for lung cancer. But if we look across all FDA approvals, you can see in the past 20 years, we've seen a significant uptick in the number of precision oncology therapies that have been approved. And you can see in blue the associated biomarkers that are specified as eligibility for those FDA drug approvals. And you can see in the last 5 years, the number of approved therapies has expanded, and this continues to be the case. So more and more targeted therapies are becoming available for patients with advanced cancers. And why this is so important, this is data taken from a cohort of non-small cell lung cancer patients, is that it's been shown across many different tumor types that patients who receive targeted therapy have better overall survival. This noted that in patients who receive targeted therapy, their survival probability was a year longer than patients who did not have targeted therapy. And again, these kind of survival data have been proven across many different cancers. So it really is increasing survival for patients with advanced cancer. All right, how about another poll everywhere question? So in a patient with a newly diagnosed colon cancer, the testing determined that it had microsatellite instability or was MSI high. So which of the following treatment strategies would be indicated for this status? All right, that's great. So far, we have 100% for initiation of immunotherapy. And immunotherapy is one of those, or when we talk about DNA mismatch repair, again, this is a different process within the DNA that now has become a biomarker for a certain type or immunotherapy specifically. So what mismatch repair does during DNA replication, we know that errors can recur. And when these errors occur, this protein complex is recruited and serves to repair those DNA changes. But when these proteins that are recruited become inactivated in some way, they fail to repair these errors, and this can cause these satellite regions or these repetitive DNA sequences to start to expand. And this on the right, you can see that in the profile of here's what a normal these repetitive sequence look like. And in a tumor that doesn't have the ability to repair these errors, you start to see the expansion of these green peaks. And this is how we can determine that a tumor is microsatellite unstable. And it's important to identify microsatellite unstable or deficient mismatch repair tumors, as the mechanism of these is that they create new neoantigens that can be presented on the tumor cell and hopefully recognized by our own innate immune system, and can then our immune system can attack the tumor cells. But there's one caveat in that the tumor cells also express PD-L1, which serves to inhibit our immune system. So novel agents called immune therapies are tumor agnostic approval that now serves to block that interaction and essentially reactivate our immune system to attack tumor cells that have demonstrated microsatellite instability. So in 2017, this was the first approval of a tumor agnostic therapy for adult and pediatric patients with unresectable or metastatic tumors that had this MSI high marker. In addition to immunotherapies, we've also seen advancements of other tumor agnostic therapies. So in-track fusions, while they're rare in individual tumors, they have a targeted therapy associated with them. So any tumor identified with an in-track fusion becomes eligible. BRAF V600E therapies are now available for metastatic tumors with that mutation, and as well as RET fusion positive tumors. So this data just indicates that it's important to identify all these rare things as there's associated therapies that have been approved by the FDA. So I'd just like to end with the success story, and that's chronic myeloid leukemia. So back in the 1960s, the chromosome rearrangement 922 was identified by a researcher. And it wasn't until decades later that we learned that this translocation of genomic material from chromosome 9 to chromosome 22 led to the creation of an oncogenic BCR-ABL fusion protein that created a constitutively active tyrosine kinase. A phase 1 clinical trial was then performed on a targeted therapy that essentially blocked the active region in the oncogenic fusion protein, and patients had a significant response. So imatinib, or the drug that targets CML cells, was the first FDA-approved targeted therapy. And now we have a number of different molecular techniques that we can use to diagnose this disease, and as well as monitor it with a RNA-based transcription and be able to determine if the disease is recurring. So in summary, I would just like to hopefully show that precision medicine is important for oncology treatment strategies, and there's more therapies on the way. That genomic profiling can be performed on tissue or liquid biopsy, and there's many different methods to do this, but by increasing detection of molecular biomarkers, more targeted therapies are approved and more opportunity to improve patient outcomes.
Right. Dr. Ramkissoon, thank you so much. And we already have our first question. And does IHC correlate with copy number gains found on sequencing tests?
I would say it depends. Protein expression by IHC doesn't necessarily correlate with copy number gains. You can see positive IHC results with just a normal copy number of that protein. So increased expression by IHC does not always correlate with copy number gains.
All right. And thank you. While we wait for other questions to come in, I had a couple. I know you were talking about the comprehensive genetic profiling and the 11 steps involved in that. That looked very time-consuming, very expensive. We hear, so as we look at efficiencies, we hear so much about the role of AI. Is there a role that's already being utilized or that's being anticipated for the utilization of AI in this process?
I think AI is becoming increasingly important in understanding how the combination of variants contributes to different tumor types. AI is becoming increasingly important in even what it can see in a tumor compared to what our human eye can see. So I anticipate a huge impact of AI on how variants can change a protein and then how that can contribute to the oncogenic process.
Great. Thank you so much. Sort of along those same lines, I know, let's see, let me look at my notes for a second here. You talked about, the growing number of guidelines. I think there were 800 there. How quickly is that growing, and what mechanisms are available for clinicians to manage what sounds like a lot of guidelines that are coming out?
There are. It's evolving all the time. And I think the NCCN guidelines are updated fairly regularly. I think if you have questions about whether something's been updated or whether a drug has been approved for a certain indication, I think that's where consulting with molecular pathologists, laboratory professionals is the best way to know for sure, just in that we are constantly monitoring or thinking about what needs to be associated with different variants. And every time we do variant classification, we're looking to see, have the indications or has the association with targeted therapies changed? So it's a constant search for information, and they're coming all the time from different agencies that are offering recommendations.
Great. Thank you so much. Let me see if we've got any other questions. I think that is it for questions today. And I know that we're a little after the hour and we do have people who need to move on. I'm sorry, can you pass that keyboard? And I'll go ahead and advance this. Thank you.
Okay.
And so I do want to say thank you to the University Cancer Research Fund for the funding that we receive and the UNC Lineberger Comprehensive Cancer Center. We want to thank the team, Benny Obore, John Powell, Oliver Marth, Andrew Dodgson, Patrick Muscarella, Lindsay Reich, and Barbara Walsh. They work hard on each one of these and they all play a role in making sure that these happen.